Hello students, welcome to EPG Pathshala. I am Professor Ramesh Kothari from Saurashtra University, Rajkot, Gujarat. Welcome you all. Today we are going to discuss about a module glycolysis under a paper carbohydrate metabolism. So students, let us see what we are going to learn in this module. We will learn we will try to understand how the glycolytic pathways is used to convert glucose into pyruvate, to understand conservation of chemical potential energy in form of ATP and NADH, and we will try to learn that what are the intermediates, what are the enzymes, and what are the cofactors are part of the glycolytic pathways. Glycolysis is an almost universal pathway of glucose catabolism, the pathway with the largest flux of carbon in the most cell. Glycolysis pathway is virtually common to all cells, both in prokaryotes as well as in eukaryotes. It is carried out in the cytosol of the cell. It is unique because the pathways can occur in aerobic as well as anaerobic condition, and hence it is considered to be the most ancient and important pathway for extracting energy from the nutrient source because primitive atmospheric lacks of oxygen. Glycolysis is the sequence of reactions that metabolize one molecule of glucose into two molecules of pyruvate with concomitant net production of two molecules of ATP. This pathway can be thought of as comprising three stages. Stage 1, which is the conversion of glucose into fructose 1,6-bisphosphate, consists of three steps of phosphorylation, an isomerization, and a second phosphorylation reactions. The strategy of these initial steps in glycolysis is to trap the glucose in the cell and to form a compound that can be readily cleaved into phosphorylated 3-carbon units. Stage 2 is cleavage of fructose 1,6-bisphosphate into two 3-carbon fragments. These resulting 3-carbon units are readily interconvertible, and in stage 3, ATP is harvested when the 3-carbon fragments are oxidized to pyruvate. Diagram shows the concept map of glycolysis. There are 10 reactions which consist of two stages. Stage 1, it is known as ATP investment stage, and stage 2 is known as ATP earning stage. Glycolysis is a Greek word. Glyx means sweet and lysis means splitting. Diagram shows the reactions that take place during glycolysis. In glycolysis, each glucose molecule split and converted into 3-carbon unit, that is pyruvate, by sequential reaction. During this sequential reaction of glycolysis, some of the free energy released from glucose is converted in form of adenosine triphosphate and NADH. Glycolysis is an almost universal central pathway of glucose catabolism, the pathway with the largest flux of carbon in most cells. The glycolytic breakdown of glucose is the sole source of metabolic energy in some mammal tissue and cell types. In anaerobic organism, pyruvate is converted to some product like ethanol, lactic acid by using fermentation. Aerobic organisms such as plant and animal oxidize pyruvate to form carbon dioxide and water. The breakdown of the 6-carbon glucose into 2 molecules of the 3-carbon pyruvate occurs during 10 steps. There are several reactions of glycolytic pathways. Stage 1, reaction 1, synthesis of glucose 6-phosphate from glucose. This reaction is a kinase reaction. Added a phosphate to glucose intermediate when glucose enter in the cell. Due to this phosphorylation, glucose transport out of the cell prevent and reactivity of oxygen is also increased. The phosphorylation of glucose in all cell in body is catalyzed by several enzymes called dehexokinases. ATP is complexed with magnesium ion, which is a co-substrate in this reaction. The reaction is essentially irreversible and the glucose is efficiently trapped inside the cell as phosphorylated intermediates do not readily pass through cell membrane. This reaction is catalyzed by enzyme hexokinase present in virtually extrahepatic cell, has a high affinity with low Km for glucose, so phosphorylates essentially all the glucose that enter cell, maintaining a large glucose gradient. This is reaction 2 of stage 1, conversion of glucose 6-phosphate to fructose 6-phosphate. The reaction is catalyzed by phosphoglucoisomerase. This enzymatic step prepares the first chiral C1 for phosphorylation. It is freely reversible reaction controlled by substrate product level. Small change in standard free energy. The reaction proceeds readily in either direction and require magnesium. This is reaction 3 of stage 1, fructose 6-phosphate to 1,6-diphosphate. The reaction is catalyzed by phosphofructokinase. The reaction is essentially irreversible. In this reaction of glycolysis, phosphofructokinase 1 is catalyzed the transfer of phosphoryl group from ATP to fructose 6-phosphate to yield fructose 1,6-bisphosphate. Phosphofructokinase is the rate-limiting enzyme of glycolysis in most tissue. It is the major regulatory enzymes of glycolytic pathway. This is reaction 4 of stage 1, fructose 1,6-bisphosphate to dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. This reaction completes the first stage of glycolysis. It is catalyzed by aldolase. The enzyme fructose 1,6-bisphosphate aldolase, often called simply aldolase, catalyze a reversible aldol condensation. Fructose 1,6-bisphosphate is cleaved to yield two different thiose phosphate, glyceraldehyde 3-phosphate, and an aldose and dihydroxyacetone phosphate, a ketose. It is an energetically unfavorable reaction in the direction written with standard free energy change, but the rapid conversion of glyceraldehyde 3-phosphate to pyruvate drives the reaction. All of the carbon of glucose can end up a pyruvate because of the equilibrium between dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, catalyzed by triose phosphate isomerase. As glyceraldehyde 3-phosphate is utilized by subsequent reaction of glycolysis, carbon is drawn from dihydroxyacetone phosphate to form glyceraldehyde 3-phosphate. This is reaction 5 of stage 1, the interconversion of triose phosphate. Only one of the two triose phosphate formed by aldolase, glyceraldehyde 3-phosphate, can be directly degraded in the subsequent step of glycolysis. The other product of dihydroxyacetone phosphate is rapidly converted to glyceraldehyde 3-phosphate by fifth enzyme triose phosphate isomerase. This is the first reaction of stage 2 and overall reaction 6 of glycolysis, that is oxidation of glyceraldehyde 3-phosphate. The reaction catalyzed by glyceraldehyde 3-phosphate dehydrogenase requires nicotinamide adenine dinucleotide, that is NAD, as an electron carrier. In its oxidized form, NADH bind tightly to the enzyme. Aldehyde group is dehydrogenated to an acyl phosphate, as aldehyde group has a very high standard free energy of hydrolysis, that is minus 49.3 kJ per mole. Glyceraldehyde 3-phosphate dehydrogenase is inhibited by iodoacetate. In this reaction, the phosphorylation occurs at the expense of inorganic phosphate. It is an example of substrate level phosphorylation. This reaction generates a high energy phosphate bond in 1.3 DGP, which is a mixed anhydride of phosphoric acid and a carboxyl acid. Because of this, 1,3-DPG has a high group of transfer potential. This is reaction 7 of glycolysis and reaction 2 of stage 2, phosphoryl group transfer. The reaction is catalyzed by phosphoglycerate kinase. This is the first step in the glycolysis that generate ATP. It is another of substrate level phosphorylation. In the prior step, two molecules of 1,3-DPG were formed from each molecule of glucose. Therefore, two ATP molecules are now formed per original molecule of glucose. Because up to triose formation, that is reaction 4, two molecules of ATP have been utilized per molecules of glucose consumed, the balance sheet for ATP utilization and formation is even at high step. The molecular structure of phosphoglycerate kinase is similar to hexokinase in that it has two lobes that each bind one of the substrate, that is ADP magnesium or 1,3-bisphosphoglycerate, leading to a large conformational change in the enzyme that brings the substrate together and exclude water molecules from the active site. It is the bioenergetics of reaction 7. The bioenergetics of reaction 7 emphasize two important concepts. First, reaction 6 and reaction 7 are coupled reaction in that the large change in standard free energy of reaction 7, that is 18.9 kJ per mole, pull the less favorable reaction 6, that is with 6.3 kJ per mole, to the right through the shared intermediate 1,3-bisphosphoglycerate as shown in figure. Second, the actual change in free energy for each of these two reactions is very close to 0, and therefore both reactions are in fact reversible inside the cell. Again, this differences in standard free energy and change in standard free energy is due to the mass action ratio, which takes into account the actual concentrations of substrate and product that exist in the cell. The reversibility of two reactions is important because when flux through gluconeogenesis is high, these two glycolytic reactions can be reversed and thus quickly respond to changing condition in the cell. This is reaction 8 of stage 2, the interconversion of 3-phosphoglycerate and 2-phosphoglycerate. The reaction is catalyzed by enzyme phosphoglycerate mutase. This reversible reaction has change in standard free energy of 1.1 kilojoule per mole. This reaction is to generate a compound 2-phosphoglycerate that can be converted to phosphoenol pyruvate in the next reaction in preparation for a second substrate level phosphorylation that generates ATP earning in step 10. It is the again reaction 8 of stage 2, the interconversion of 3-phosphoglycerate and 2-phosphoglycerate. The metabolism of this highly reversible reaction is illustrated in above figure, where it can be seen to require a phosphoryl transfer from phosphorylated histidine residue located in enzyme active site, which is phosphorylated by transfer of phosphoryl group. In step 1, the substrate 3-phosphoglycerate bind to the enzyme active site and is phosphorylated in the carbon 2 position by a transfer reaction involving the histidine phosphate group. This type of substrate interaction with the enzyme is non-covalent and referred to as a substrate-enzyme complex. The short-lived intermediate 2,3-bisphosphoglycerate is created by phosphoryl transfer from the histidine residue to carbon 2 atom of the 3-phosphoglycerate. In the second step of the reaction, the C3-phosphate is transferred back to the histidine residue of the enzyme to regenerate histidine phosphate, leading to the release of 2-phosphoglycerate and binding a new molecule of 3-phosphoglycerate in the third step. Note that the 2,3-bisphosphoglycerate, which is also called BPG, formed in step 1 can diffuse out of the active site, resulting in dephosphorylated enzyme, and you may remember that blood cells BPG has an important role in regarding oxygen binding to hemoglobin in red blood cells. When BPG leaves the active site without rephosphorylation, the histidine group of the enzyme can only be activated when trace amount of BPG diffuse back into the active site. This is reaction 9 of stage 2, dehydration of 2-phosphoglycerate. In this reversible reaction, water molecule is removed from phosphoglycerate to yield phosphoenol pyruvate is prompted by enolase. In this step of glycolysis, 2-phosphoglycerate is converted into phosphoenol pyruvate by dehydration reaction, which is catalyzed by enzyme enolase. It is interesting that the change in standard free energy for this reaction is relatively small, that is 1.7 kilojoule per mole, meaning that the overall metabolic energy available from 2-phosphoglycerate and phosphoenol pyruvate is similar. However, when enolase convert 2-phosphoglycerate to phosphoenol pyruvate, it traps the phosphate group in an unstable enol form, resulting in a dramatic increase in the phosphoryl transfer potential of the triose sugar. The standard free energy change for phosphate hydrolysis in 2-phosphoglycerate is 16 kilojoule per mole, whereas for phosphoenol pyruvate it is an incredible, that is 62 kilojoule per mole. This is reaction 10 of stage 2, synthesis of pyruvate. This last step in glycolysis is catalyzed by enzyme pyruvate kinase. the formation of ATP from ADP at the expense of high energy phosphoenol pyruvate, phosphoenol bond of phosphoenol pyruvate. This step is also an important site of regulation. In this reaction, the high phosphoryl transfer potential of phosphoenol pyruvate is used by 3-enzyme pyruvate kinase to generate pyruvate, the end product of glycolysis, and 2-adenosine triphosphate are formed for every glucose molecule entering the pathway. This is the second of two substrate level phosphorylation reaction in glycolysis that couples energy release from phosphate hydrolysis, that is 62 kJ per mole, to that of ATP synthesis, which is 30.5 kJ per mole. Pyruvate is a stable compound in cell that is utilized by many other metabolic pathways. The ATP investment phase of glycolysis include the energy conserving phosphorylation step, in which some of the free energy of glucose molecule is conserved in form of ATP. The second stage of glycolysis begins with the splitting of fructose 1,6-bisphosphate into glyceraldehyde 3-phosphate, which is also known as GAP, and dihydroxyacetone phosphate, that is DHEAP. The product of the remaining steps in glycolysis consists of 3 carbon units rather than 6 carbon units. Glyceraldehyde 3-phosphate is one of the direct pathway of glycolysis, whereas dihydroxyacetone phosphate is not. Following is the overall balance sheet net gain of ATP during glycolysis. Under aerobic condition, the two molecules of NADH are reoxidized to NAD plus by transfer of their electron to the respiratory chain in the mitochondria. So, dear students, remember that one molecule of glucose yield 2 molecules of glyceraldehyde 3-phosphate. Both halves of the glucose molecule follow the same pathways in the second phase of glycolysis. The conversion of 2 molecule of glyceraldehyde 3-phosphate to 2 molecule of pyruvate is accompanied by the formation of 4 molecule of ATP from ADP. However, the net yield of ATP per molecule of glucose degraded is only 2, because 2 ATP were invested in preparatory phase of glycolysis to phosphorylate the 2 ends of exhaust molecule. So students, let us summarize what we have learned in this module. Glycolysis was the first completely elucidated, and it is the most extensively studied pathways. Glycolysis is a ubiquitous pathway. All living cells contains some form of glycolytic pathway. This implies that glycolysis is a very old pathway, one of the first evolved, and it suggests that the breakdown of glucose sugars for energy is important for all type of life processes. It is the only metabolic pathway in mammalian cell that can generate energy in form of ATP. In the absence of molecular oxygen, it is an anaerobic pathway. It is the only energy generating catabolic pathways that occurs in cytoplasm of eukaryotic cell. The other energy producing pathways occurs in the mitochondria. Glycolysis exhibit many of the underlying themes of metabolism. So this is all about glycolysis. Thank you very much.